Hi everyone, and welcome to the third ABL seminar. Today we have the pleasure to receive Julio Cesar Bicca-Marques and two of his former students, and I will start by shortly present them. Dr. Julio Cesar Bicca-Marques received his undergraduate degree in biological sciences from the University of Rio Grande do Sul in 1986 in Brazil, where he went on to receive his master's degrees in ecology from the University de Brasilia, and then a PhD in biological anthropology from the University of Illinois at Urbana-Champaign, where he remains a research associate to this day. He is currently a professor at PUC de Rio Grande do Sul in Brazil to this day. He is currently a professor, where he is also the head of undergraduate research and a member of the graduate program in ecology, evolution, and biodiversity. His lab currently studies behavior of various non-human primates. Joining him are his two former students, Thalita Siqueira Sacramento, a former PhD student who studied social foraging strategy in marmoset, and Karin Galisteo Diemel Lopes, a former master's and undergraduate student who studied environmental factors influencing behavior of howler and tinty monkeys. Today, they will give a talk entitled Studying the Behavior of Free-Ranging Brazilian Non-Human Primates. Thank you for being here, and you can start when you are ready.
Okay, thank you, Alexis. I'd like to thank you, Amanda, Amelia, Abby, Samadi, Natasha, Matilda, Syed, and Valentin for the invitation to talk with you today. And first of all, I'd like to say that Brazil is the #1 country in terms of primate diversity. So we are in a very good position to study non-human primates. And at the Laboratory of Primatology at PUC in Rio Grande do Sul, Brazil, we studied the behavior, ecology, conservation, and cognition of non-human primates with a focus on our Brazilian ones, particularly the primates that live in our state of Rio Grande do Sul, but also we have been developing studies in all major Brazilian biomes. And so we have been covered several species and genera. I'll present you some of my major research topics. The first one is the cognitive ecology. That means that we are looking for to identify the environmental information that primates use in their search for food. And for doing this, we run experimental field studies. I'll show you later that in our other research, we make mainly observational studies with free-ranging animals and using different research methods. But for doing the experimental studies that we do in the cognitive ecology and social foraging that sometimes we run together, we need to trap the monkeys. And normally we have been doing this mostly with callitrichines that are very small New World monkeys. And for trapping them, those that we can trap, because we have some studies with howler monkeys and also capuchin monkeys that are much more difficult to trap, and we didn't trap. We use for trapping marmosets and tamarins, we use the Saguinas trap. That's a method developed in Peru, and that you can see here, we built this trap with 10 separated cages where we can, from a blind, control the strings to close with each individual inside. And so it's a very effective method for trapping them safely. And then we trap them to mark them with color-coded collars that we can see, and sometimes also with some kind of trichotomy in the tail mostly, so that we can identify each individual while they are visiting our experimental field stations, feeding stations, and so that we can understand the behavior of each individual. Here we have in the top left, the Leontocebus weddelli. It's the current scientific name of this species. It's a tamarin. It's a saddleback tamarin. The emperor tamarin, one of the most beautiful ones, it's the Saguinus imperator, and one titi monkey. It's Plecturocebus topitni right now. The names have been changing in the last years. And here on the bottom, the two photos of individuals of both the saddleback tamarins and an emperor tamarin visiting a feeding platform. So we built a feeding station, artificial feeding station or more. In my PhD thesis, I worked with that. We had 4 feeding stations. I had 3 people helping me, assistants. I'll show you that each feeding station was composed of 4 feeding platforms. And we observed the monkeys from a blind, from inside the blind, in order not to disturb the monkeys with our presence. So they knew that there was someone inside, but we were covered. We had this green cover in the front of the blind. And so we are kind of really hiding inside the blind. And we could take notes from what they were doing. We could record the data from inside the blind. Our platforms could be reward platforms, those that contained bananas as a bait, and also non-reward platforms that, depending on the experiment, I used to use plastic bananas that looked like real bananas, especially some real bananas, and that they would need to learn that they weren't actual bananas, and they actually tried to bite them before understanding that those perfect bananas were perfect because they weren't bananas. And also we used to use real bananas inside the wire mesh cages that I made myself as the false leaves that you can see here on the platforms. And that served to control the release of the release of olfactory cues from the platforms, from the non-reward platforms. When I didn't want to have olfactory cues, then I had real bananas that are inaccessible inside wire mesh cages. And so our feeding stations were composed of 8 feeding platforms, and in part of the experiments that I'm gonna show you some results in a moment, we had only 2 of them baited with real bananas, or reward available bananas at least. This gives us a chance level of 25% that the monkeys would be able to find at random the real rewards. And so performances above 25%, depending on the number of visits, would be them considered significantly above chance level, and that they had learned to use that cue to find the food. We have tested spatial information. We did that by keeping the same reward platforms throughout the experiment. And so once they visited the station and found the food, they should use a rule, win return rule or win stay rule. That means that if I ate here before, I can come back. If I am eating now, I can come back later and eat again at this platform. And when we tested this with several species, including night monkeys, that I'll show you also a difference in the use of visual and olfactory cues, then they all performed very well. They learned very quickly that the platforms that contain the food rewards, and they tend to return straight to those platforms in future visits. And here is from a paper that they published on the win-stay rule, where you can see the performance of titi monkeys that here to the left place was predictable, and they almost always returned straight to the reward platforms. When place wasn't predictable, they were no longer predictable because I was testing, or it was a control experiment, or I was testing olfactory cues, visual cues, and other cues that where I needed to manage the distribution so that the platforms were randomly, the reward platforms were randomly defined at each session. They stopped using the win-stay rule. This is the expected near 25%, and they did actually vary their performance. Once I resumed using the spatial information constant in an experiment previous to my final one that measured the use of the amount of food in the platform as the cue that they needed to use. I had like a half banana against the 3 bananas in one and the other. And then they quickly began to use again the win return or win stay rule. And the marmosets did very well as well. When it comes to visual cues that I had uncovered rewards, the place of the rewards, the reward platforms and non-reward platforms varied from one session to another, but the bait was exposed to the monkeys. Diurnal monkeys did much better than nocturnal ones. The night monkeys had a harder time to learn the difference and to differentiate the bananas, and so they took longer to learn this information. Night monkeys are monochromats. They see like in one color. They have a low reception in the red wave of the color gradient. And so I had some 35 watts lamps to have the platforms, all platforms illuminated so that I could see, because it was, even this way, it was very hard doing this at night in the middle of the forest. And all other diurnal primates did much better. Howler monkeys that are this one, they are trichromats. They are the only New World monkeys that are trichromats like ourselves, that's seen 3 basic colors. And all the others that we have here, mostly the calitrichines, they have a color vision polymorphism where males are always dichromats in 3 kinds of dichromacy, like daltonic people. And females can be either trichromats in 3 kinds also, or dichromats. So they have a different way of seeing the world. It's a very interesting aspect. But all diurnal monkeys had a much better performance than the night monkeys. On the contrary, when we controlled for this location, we covered the bait that the non-reward platforms had a fake banana under it, so that the only platform that was releasing banana smell were the reward platforms that had a real natural banana under the false leaf. Then in this experiment, night monkeys performed much better. That's compatible with their lifestyle. So I'll move to social foraging. This is a study that I'm only going to show you the experimental setting here. That's a little different from what I have made in my own study. We had in this experiment, there's a collaboration with colleagues from another university, Maria Fernanda, who's the first author. She was my co-advised student in a PhD student. And we studied something similar in terms of the information that they were using, but also how the individuals would behave in the feeding station using the social information, behaving either as producers, those that arrive first in the each feeding platform, or the scroungers, those that join others and try to steal food from them, or at least co-share the platform with them. We had boxes where the marmosets learned to insert their hands. And one very interesting result that was an expected result, particularly based on in data that we had from the literature from captive studies, and also from my own PhD study, where I saw that adult females, the dominant adult females that are the ones that normally breed, the others normally don't breed, only one adult female. She monopolizes the breeding in each group, the dominant female. And Fernanda observed here also that independent on the conditions, if we had more food scattered or less food scattered, or more food concentrated or less concentrated, the adult dominant female, that is this one, always had a better, or in general, had a higher food intake than all other individuals. And so this is something very interesting. I'll now show you a video that Talita recorded, because she ended not being able to be here in person, and so she recorded her presentation for us.
Okay. Good afternoon. I am Talita. I was a PhD student at Bicca-Marques University here with Professor Bicca-Marques as my advisor. So today I will present some results of our research that entitled Scrounging Marmosets More Than the Final Share is Good Without Changing Their Searching Imports. Generally, animals that live in groups end up searching for food together, a situation that brings different challenges than forage alone. Foragers may rely on social in addition to ecological information to locate feeding sites and choose between different foraging strategies. A number of social foraging models have been proposed based on game theory. One of them is a producer-scrounger model, in which different individuals may behave as producer and spend time and energy using ecological information to actively search for food, or as scroungers and focus their foraging efforts on finders' behavior and scrounge part of the food they find. One factor that may influence the choice of foraging strategy is the finder share, defined as the proportion of the food resource consumed by the producer before the arrival of the scroungers. A number of models predict that when the finder share is relatively small, the frequency of scrounging will be high, because producing gives only a small benefit compared to playing scrounging. A model based on linear operator learning rule confirmed this prediction and further predict that individuals would change their foraging strategy gradually in response to environmental changes. We tested these predictions in an experiment involving 3 groups of free-ranging black-tufted marmosets, Callicrix pedicillata. We captured and marked all individuals. We manipulate the find the shed at the artificial feeding stations by controlling the distribution of the food rewards, slice of banana, among the baited platforms. We divided our field experiments into two conditions: low find the shed, few food rich patches, and high find the shed, many food full patches. Used Excel functions to randomize across sections which platforms containing accessible food. Now we will watch a video from one section. So we recorded the number of times and frequency that the focal individual played producer, that is, arrived at an unoccupied platform, and the number of times that it played scrounger, that is, arrived at a platform occupied by another marmoset. We recorded a new event when a marmoset re-encountered an unoccupied platform or re-scrounged an occupied one.
Sorry, it seems that you weren't listening. Is that right?
Yeah, sorry, Julio. Apparently, the sound is really low for people on the live.
Okay.
And apparently, when you are muting your mic, it's also muting the video.
Yeah.
So there is no real solution.
Well,
maybe what we can do is just go to Karin, and maybe after you can open the full video just to see at the end if we manage to get the sound correct.
Yes, okay. I'm going forward here with my part, and then Karine can talk hers, and then we can see if we can watch Talita. If not, I can synthesize what she's presenting for you without
thank you.
the slides. Okay.
Perfect. Thanks. Sorry for that.
So let me see if this is.
We also recorded the number of visitors.
No, I don't know what's happening. I'm not being able to stop this.
Maybe just escape.
I'm trying.
Oh,
it didn't
work.
Okay, now it worked, but then I'll move here. Okay, I'm back. And so the third research line that we run in my lab is one that is the most important, that we have more publications and more studies on, is on the effects of habitat restriction on primate behavior and ecology. And so these kinds of studies, we have focused on several species, although mostly howler monkeys, many studies in our state of Rio Grande do Sul, here in south of Brazil, the southernmost distribution of primates in the Americas. And these are observational studies that in which we habituate the monkeys to our presence first, then we follow them from dawn to dusk for several days. Sometimes for the least has been 3 days, but some 4, normally 5 days per month, and sometimes even 15 days per month. And so it's very Well, it's a time consuming. It's you need to be tough to do this for many days in a row, for many months in a row, and but it's wonderful. It's something like it's really very nice to be around with the monkeys and understand how they behave. And we normally use, most of my studies have been focused. Sorry, there's someone doing something upstairs, and some of our studies have been used scan sampling method, but a few students have used a focal animal method of data collection. I'm really sorry for that. It wasn't expected to have my neighbor doing something in her apartment. Our studies have focused mostly on howler monkeys, and they are capable of living in very small forest fragments or sometimes even orchards. This is an orchard here that has something like 0.6 hectares. It means that's a square of 60 meters by 100 meters, something like that. And they are capable of surviving there. I'm going to show you how they do that and how they are adapted, how flexible they are in some aspects of their ecology and behavior, but also that they are capable of doing this as individuals, and but as populations, this isn't that safe in the long term. So this is very important. It's a message that I want to show you. And this is the black and gold howler monkey, the adult male and the subadult female. Well, this line, I started this line during my master's when I studied in a small orchard, like that's larger than that one, but it's a small orchard. And I found several very interesting patterns that I'll show you in a moment. And then in 2002, I was invited to write a chapter for this book, the first book in Primates and Fragments, edited by Laura Marsh. And then I conducted a literature review to understand how do howler monkeys cope with habitat fragmentation, actually habitat restriction or habitat shrinking. That's how they would behave. I compared groups living in forest fragments or continuous forests of different sizes. And so trying to understand if those that used to be in smaller and shrunk habitat patches, if they would behave differently. What I found was very interesting. Well, first finding, and an expected finding, that the home range tended to be smaller when the size of the fragment was small. I also found weak tendencies that when a direct relationship between fragment size and the number of plant species exploited as food sources, also the number of sources of leaves and the number of sources of fruit. All other aspects that I studied in terms of day range and other aspects of terms of the activity budget, they didn't change. Later in 2015, we published another book chapter. Now, the leading author was my previous PhD student, Vanessa Barbisa Fortes, and Thiago had been my master's student. And we found here that fragment size in this analysis did not predict home range and day range. So it confirmed my results, previous results on day range, irrespective of the size of the home range and the size of the forest fragment. They normally do not travel more than 1,000 meters per day. Normally, the median ranges, the mean ranges between 600 and 800 meters. So they are very calm, they rest a lot, and they range not that much each day compared to other attalids, that is the families to which howler monkeys belong in. In another study, that is small 0.6 fragments, we found that the average temperature had an important influence on day range. The highest temperature, more they traveled more. Travel is quite an exaggeration, considering that the site was very small, but they walked and covered longer distances when it was hotter than when it was colder. And this was made by a previous student of mine, Elisandra, and with the help, we analyzed with the help of my previous master's and current doctoral student, Gabriela. And then I got a very good grant here in Brazil to conduct a 5-year study that ended being much longer with Oscar Chavez. It's a Costa Rican guy that came here, a very nice collaborator and friend. He's no longer here now, but he did a very nice job. But we had 6 forest fragments. We compared 3 large fragments near 100 hectares and 3 groups living in small fragments smaller than 10 hectares. And the idea was to compare their ecology and behavior to see if the patterns that I had found in my previous reviews were also observed once we had the groups of the same species living in distinct conditions in terms of habitat shrinking, but in the same kind of forest. And so we did this study here in Porto Alegre and Viamão. And I'll show you some of the results. This one comparing their diets. We can see that the intermonth variability in terms of the similarity of the diet tend to be lower in the large fragments. They varied more from one month to another, their diet, than the groups that were living in small fragments. But we found what we expected, that the contribution of different kinds of foods like mature leaves, young leaves, ripe fruit, and unripe fruit are the most important foods that howlers eat. They were quite similar, and also the only difference that we found, a strong tendency, was that the monkeys live in large fragments. They explored more palms than those living in small fragments, whereas trees, lianas, and vines had similar contributions. In my master's study, together with my wife, Claudia Bicca-Marques, we had published one of the first studies showing that the animals lived in this 2 hectares orchard. We found that the exotic plant species played a very important role in the diet of our study group. From 27 plant species that they explored, 11 were alien species, and they contributed to 38% of their annual diet. And so we wrote this paper in 1994, calling the attention of the scientific community of the importance of exotic plant species as staple food sources. And we also suggested that we should consider using alien species, non-invasive, that's possible, alien species, to promote the recovery of forests and to promote even maybe stepping stones of between habitat patches, so that the monkeys could disperse more easily in this anthropogenic landscape. More recently, I was invited to contribute to a special issue on crop feeding, and then Oscar and I did this analysis on the data that we had used. They are all, these cultivated ones are alien species as well, and they were only present in our study here, this with 6 groups in the small fragments. And the interesting finding here was that the use of cultivated species wasn't related to the availability of native plant species, native fruit, but was significantly related to the availability of decultivated fruit. So this means that the monkeys, the howler monkeys, were monitoring the availability of the cultivated fruit and were preferring to go there to eat because they are normally much more, they are juicier than the native fruits that they normally have available in the large fragments. And so it's an interesting finding. I was also one of the first to publish this is 1992, to publish a paper on the drinking behavior of howler monkeys in my master's, and I didn't find the evidence of drinking in terms of a detoxification strategy based on what they were eating in terms of leaves and fruit during each month, but there was a significant relationship between the use of 3 reservoirs of water and rainfall. That's obvious. When we have more rainfall, the 3 reservoirs would be filled up with water, and then tended to drink more water from that source in that month. We did then recently just published a paper on drinking behavior with 14 groups, 6 studied by Oscar, 4 studied by Vanessa, 3 studied by Gabriela, and 1 is studied by Renata. It was also a former student of mine. And we also confirmed our previous findings that there's no difference between, it can't be predictable based on fragment size. It all depends on what kind of source of water they have available, and they tend to behave similarly, irrespective if they are in a larger, small, or a medium-sized fragment. And we also found that flower consumption, ambient temperature, and rainfall modulated their drinking behavior. In another study of master students, Naina, that's doing a continuation of this study now in her PhD, adding also analysis of microbiome and other information related to the health of the howler monkeys. But she studied the influence of food supplementation, people that used to feed the monkeys that live around their homes, on their diet and activity budget. What she found was that once the monkeys were eating the supplements, that 1 of 2 groups actually, 1 group only received fruit, and the other group received fruit, bread, and other things that they shouldn't be eating. But both groups decreased ingestion of wild fruits when they ate more greater biomass of supplemented foods, but they didn't change their activity budgets. There is the rested, fed, moved, and socialized similarly, irrespective of the amount of supplements that they were eating each day, and they did not also change their ingestion of wild leaves. And this confirms our finding from the crop feeding study that they do like the juiciest fruits that they have available when they visit the orchards around homes, wild, free-ranging and more wild groups that Oscar has studied, but also these groups that are not domesticated, but they are more habituated to people, and the group that used to eat bread was fed by hand. So the other was in a platform also. And we have also recorded in the picture here was from my master's student and a subadult male black and gold howler monkey going to inspect a oven bird nest. He didn't uh, inserted his hand because the bird was around and chased him away. But in another study, Karina was a former master student of mine, studied a group in a very small uh, forest, and she saw the monkeys eating eggs, chicken eggs. They used to go inside the place, or the rooster, where the place where the chicken are are kept, and to steal eggs for eating. And so we published this paper on habitat impoverishment and egg predation as a potential behavior that howler monkeys adopt when they have a restriction in terms of diet and field diverse of leaves in terms of protein and so on. And later, we then conducted here near Porto Alegre with another species that they had a report that they also used to eat eggs. And so we ran in 7 groups experiments of artificial nests, and one group only. And the time that we worked, these 3 were my students at the time, all undergraduate students. We had one group that ate the eggs, and the others didn't show an interest. I'm close to finish this part, but with them studied also with a colleague of mine in another postdoc the stress hormones, the release of stress hormones in the feces of howler monkeys in the 6 groups that the Oscar monitored. And we found that there was no influence of fragment size on their stress level. They had the same level of release. We had a small difference between nursing females and the other females and males, but we didn't find any indication, contrary to some other studies, that our howler monkeys that lived in very small fragments were more stressed than other than the other howler monkeys. We have a hypothesis that although they were closer, near, in smaller, living in smaller home ranges, we know that the other groups, the ones living in larger fragments, they had neighbor groups, and this may have increased their level of stress, whereas those that lived in the small fragments, they were living alone in these fragments, although they have more contact with people and dogs and other challenges and threats, but they didn't show any different sign of stress. This is very interesting because then we could expect that we would find howler monkeys everywhere. And indeed, some people, even people that work with conservation, sometimes think and say that howler monkeys can be found everywhere. In fact, they can be found everywhere, almost everywhere, but they aren't found everywhere. What that means, as I told you before, they seem to cope very well at the individual level with the conditions found in small fragments, but they didn't do that well when it comes to a medium or long-term survival at the population level. And so this study done at that time by my undergrad student, Felipe N. Silva, now a doctor. He conducted this study also as an undergraduate, but then he was my master's student with another study. And we found here in a city near our capital, that many fragments, all the ones in red, weren't inhabited, although they had conditions to be inhabited by howlers. And we found here that we didn't see any relationship with fragment size, but we found that probably some historical event has wiped out the howlers from the fragments under this, to the right, and these two roads, RS 709 and BR 116. And in another study, Gabriela, that's now finishing her PhD in her master's, she studied in another region of the state, the same species. And what she found was that size mattered. Howler monkeys were found in forest fragments smaller than 8 hectares in only one of them. And so we found that the smallest fragments, although they could be inhabited by howler monkeys, they no longer were, and so they seem to be functioning as sinks in this region. So this is an important information, and this led us to publish an idea that I have been defending for many years, and we published, also invited to contribute to a symposium in the International Meeting of Primatology, and then the organizers organized an issue of the American Journal of Primatology, and so we wrote it's not a review because it was not an extensive review, but it was a compilation of information that I have been working on and reading over my almost 30 years, more than 30 years now, working with howler monkeys, that their tolerance to habitat shrinking, instead of being a lifetime warranty, as is seen by many researchers that even work with conservation, they are more like a death sentence in the long term. Why? Because populations that live in small forest fragments and that are more isolated in the landscape, they suffer from inbreeding depression, or even sometimes stochastic problems, like the group that demographic stochasticity, like the group that Karina studied. It was a group composed of an adult male, an adult female, and 3 offspring males from this couple. And so there was no new female to keep the population growing unless some female could come from another nearby inhabited site, and so they normally face several problems. And they also, in addition to problems of inbreed depression resulting from low gene flow in these situations, they suffer from dog predation, electrocution, and road kills. We are close to finish a paper to submit for publication comparing 2 regions in South Brazil with more, probably near, if I'm not wrong, 500 deaths over some years. And also in these fragments, although they can be preyed upon by dogs, as I showed you, when they try to cover the anthropogenic landscape to go from one fragment to another, or from one part of the home range to another where there's a canopy gap, also, once they are in a fragmented landscape, if there is a predator, they can be preyed upon also by wild animals, like wild predators, such as this case observed by my master's student Erica in her study in the state of Rondônia in the Brazilian Amazon with Aloata puruensis. That's the first case of a predation event by a snake on an atellid primate, that are the largest one in the neotropics. In addition to dog predation, roadkill, electrocution, we have also disease. And then I'm close to finish, because this is something very important to tell you, considering that in addition to all the threats, the monkeys can, the howler monkeys are very susceptible to the yellow fever. And together with the effect, the impact of the yellow fever on the populations, that we know now that about 80 to 90% of individuals in each population die, and sometimes the small populations that inhabit those fragments, they are wiped out from the landscape because they are very susceptible. The media sometimes delivers wrong information to lay people, and then people think that the disease is the disease of the monkey, and they start killing the monkeys. This is illegal in Brazil, but we received many reports and people saying, yes, I killed because I was scared. And then all this information and several others that I don't have time to present to you allowed me to launch a campaign. It's campaign Protect Your Guardian Angels, because howler monkeys are, and other primates as well, but howler monkeys and a few other species that are more susceptible to the yellow fever virus. Once they start dying very quickly, they signalize to the health authorities that they need to vaccinate the population. And so Davi and I, that was a former master's student of mine, wrote this paper discussing the role of monkeys, mosquitoes, and humans in the occurrence of yellow fever outbreak in a fragmented landscape in South Brazil. And the idea of my campaign is that protecting howler monkeys is a matter of public health. And I can show anybody with the data and the knowledge that we have in terms of ecology, behavior, and population ecology of howler monkeys, and this data on distribution, that it's simply impossible that the spread of the virus was the responsibility, the rapid spread of the virus in these fragmented landscapes was caused by monkeys, based on ranging behavior and many other information. And then later we conduct a study with a PhD student of mine, Marco Almeida. That's my major professor on yellow fever. He works on this for like 20 years in the Health State Secretariat. And we run a modeling of the data that they had at the Secretariat of the 2008-2009 outbreak in our state, that came from the northwestern part of the state, and in like 8 to 9 months, it covered almost 600 kilometers. And we modeled this spread and also the distribution of howler monkeys and the mosquito vectors with the help of Rafael, that was there. It was currently a PhD student at another university, but was my undergraduate student. And we found that the factors that explained the spread were air variation in relative humidity, mean annual rainfall, and maximum temperature in the hottest months, that are related to mosquito reproduction. Host distribution, obviously, because we were only analyzing data on the disease in howler monkeys, so this couldn't be different. But the fifth factor that showed an importance of 11% was maximum wind speed. That is compatible with the idea that mosquitoes were not only can fly from one forest fragments to a nearby one, sometimes even for some kilometers, but they can be taken and blown away by wind gusts. And so if we see the pattern of wind, the circulation in our region, it makes exactly the way that the virus, that the disease made in 2008, 2009 in our state. So it's very important to have this basic knowledge on the behavior and ecology of our study subjects. And it was very nice to be able to apply this knowledge to this problem that is a conservation problem, but also a public health problem. And so I'd like to thank the financial support that I have received during these years for these studies and for my students' studies as well. And so I'll pass the time to Karine present her study on also the effect of habitat shrinking on a different species in the Amazon region, hopefully without a neighbor making noise upstairs. Go ahead, Karine.
Okay. So thank you for the invitation. I'm going to talk a little bit about the results of my master's research, the response of titi monkeys to food availability fluctuations. So titi monkeys comprehend 3 genus that are very species rich. They are small primates with a wide distribution in South America. Here we can see the distribution of the 3 genus, Queracebus, Plecturocebus, and Calicebus. Titi monkeys are frugivorous primates, and they complement diet with leaves, seeds, flowers, arthropods at different proportions depending on the species. But fruits, which are the main food item for titi monkeys, are seasonally available. And so they usually face periods of food shortages. During these periods of food scarcity, primates increase the consumption of low-quality food items, which are usually difficult to process and digest because they are mechanically and chemically defended and are also poor in easy-to-digest carbohydrates. So these changes in diet may alter the activity budget of primates. As the energy intake is lower, animals may need to spend more time feeding and or resting to compensate that. And titi monkeys do that. So Calicebus melanochyr, Queracebus lugens, and Calicebus nigriformis increase the consumption of seeds when they're consuming less fruits, while Plecturocebus oenanthe increases the consumption of arthropods when they're consuming less fruits. Others, like Plecturocebus bruneus, Calicebus coimbrai, Calicebus nigriformis, increase the consumption of leaves. And as I said before, these changes in diet also promote changes in the activity budget. For example, Plecturocebus bruneus and Calicebus nigriformis adopted an energy minimizer strategy when they were consuming less fruits. So the animals reduced energy consuming activities such as feeding and traveling and spent more time resting. So this was the first study to investigate the diet and behavior of Plecturocebus bernardi. We investigated how these titi monkeys respond to food availability fluctuations, and we tested whether animals would consume food items according to its availability, would diversify diet when preferred foods are scarce, and if they would adopt an energy minimizer strategy as other TT monkeys. Our study was conducted in a small fragment in the Brazilian Amazon. The region has a tropical wet and dry climate with a dry season between June and August. Our study we followed the group from March to August 2015. We used scan sampling to record animals' behavior. And the study group was composed by 4 individuals, an adult male, adult female, a young male, and a female infant. We estimated food availability in the fragment, leaves, flowers, and fruits, collecting phenological data. We used 5 categories to do that. 0 meaning the absence of leaves, for example, in the canopy, and 4 meaning that 76 to 100% of the canopy was covered with leaves or flowers or fruits. We also estimated prey availability. We used water traps, 15 traps placed in the canopy and 15 traps on the ground. And after that, animals captured were measured and separated into big and small. And then we compared food availability, both plant parts and prey, between the rainy and the dry season. We found that adult leaves were more available during the rainy season, and that unripe fruits and also ripe fruits were more available during the dry season. And we found no differences for the other food items between the rainy and the dry season. So Plecturocebus bernardi had a frugivorous diet. They ate mostly ripe fruits, and they also complement their diets with plant parts, flowers, arthropods, seeds, and others. And seeds were consumed only during the dry season. We found a seasonal variation in diet. Animals ate more ripe fruits during the rainy season and also more arthropods. And they increased the consumption of flowers during the dry season. We found no relationship between food items and food consumption, except for large arthropods that were ingested proportionally to its availability. We found also variation in the activity budget. Animals spent more time feeding in the rainy season and also more time traveling. And they spent more time resting during the dry season. So Plecturocebus bernardi had a frugivorous diet like other TT monkeys. We found no relationship between food availability and food consumption. In fact, animals ate more fruits when this item was less available in the fragment. This was mostly because of 2 preferred species that were fruiting during the rainy season, Nectandra and Orbignia phalerata. So during the rainy season, the animals focused their feeding time on these 2 species. And although we found a relationship between large arthropods availability and consumption, it seemed more like an opportunistic behavior of the species because animals were not actively searching for prey. It seemed like they would detect and encounter more prey as they were more available. In the absence of those 2 preferred species, TT monkeys diversified their diet. They increased the consumption of flowers, and they also included seeds in their diet. And Plecturocebus bernardi adopted an energy minimizer strategy like other TT monkeys. They spent less time feeding and traveling and more time resting when the consumption of fruits was lower. So to sum up, TT monkeys responded to fluctuations in food availability, adjusting their behavior and diet. When fruits were less available in the fragment, animals focused on this item and they spent more time feeding and traveling. On the other hand, when they consumed less fruits, they diversified diet and also spent more time resting. As Bicca already said, fragmentation is something that we have to take in consideration. The group inhabited a small fragment, and in small fragments, the number of tree species drops and plant community and structure changes. So this may affect food sources for primates. Although titi monkeys, as well as howler monkeys, are able to cope with fragmentation, they can live in small fragments. It's not clear how this could affect their survival in the long term for all the reasons that Bicca already said: isolation, inbreeding, all that. It's something that we still need to study more to understand, as fragmentation is becoming more and more common here in Brazil every day. So I would like to thank Bergamin, Quintino, and Pivolio family for their support, logistic support to this research, and also all these institutions for the financial support they gave. And thank you all for your attention.
Thank you guys for this nice presentation. Julio, because like we are kind of a little bit over past the time already, what I could propose regarding the video of Talita, because I would not like that people are not able to see it. You could maybe send us the video, and we could incorporate on YouTube afterward, like this people can see it. That works for you?
Yes.
Yeah.
Yes,
and maybe you could provide an address, like these people can ask questions if they have.
Okay.
So,
okay.
Yeah.
That's
we have some question, and I'm sure that the chat will continue to ask some. So first we have Amanda, who asked like during your cognitive experiment, like that you are using some color tag, and she was wondering if the color tag could change the behavior of the monkey. So for instance, social behavior or things like this.
Well, I used I tried this in the wild with red and yellow, if I'm not wrong, but they didn't perform very well. I had one, if I'm not wrong, I never published that, so it's in my thesis only 20 years ago. And if I'm not wrong, one individual, one tamarin apparently performed above chance level, but it was some like beginning to show some use of the difference between yellow and red. It was a red with a strong hue, let's say a dark red. Mm-hmm. And in captivity, we did that with marmosets, with a PhD student that we still didn't publish also. And if I'm not wrong, also some individuals started showing some sign of the use of the visual cues, but I can't answer with certainty.
Okay, like for instance, it's not happening that like a monkey with the color tag will be aggressed by some.
Sorry. I misunderstood the question. I thought you were asking if they were using the color cues on. I mean,
in modification of the behavior.
The colors. No, this didn't influence their behavior.
No,
this was no. The problem that we could have was, if you didn't fit the color correctly, it would be loose and they would put in their mouth. And so you need to fit the color with a finger, not in infants because they would grow and they could be strangulated with the color. But for the others, probably a few hours they get used to them and are fine.
Okay. We have Natasha Rossi who says, so first, thank you both for this very interesting talk. I think the question is for you, Julia. Like, so she's asking, what will be the take-home message that you would like the audience to get in terms of species management based on your results on habitat fragmentation?
Oh, excellent question. Well, there are many things that we can make. Well, first of all, we need to understand that they are not spreaders of diseases because they are living there. They never, well, it's not common to be ranging in the landscape, so don't kill them. Second, we can, depending on the situation and the population density and the number of fragments that are inhabited, if we don't have like well, if they can't, if there isn't gene flow without the management, we may need to manage individuals and change individuals to increase gene flow even artificially. And we can, as Karine said about for the titty monkeys, that we lose the number of trees and we lose tree diversity also when the habitat becomes smaller. So we can enrich the forests with native plants that we know that serve as important food items. For example, for howler monkeys, ficus species are very important. And so if there is no ficus there, you can put ficus. We have some other plants here. There's one palm, and there's also another tree that's gymnosperm. That's the pine here in our state. It's also an important food item in some parts of the state. So you can manage and try to grow this. That would be obviously long-term strategies. They can't be short-term because they take a long time to grow. And maybe we can also start using exotic trees, as I have said, though, especially those that you know that can't be spread by the monkeys. Because we have made also studies on frugivory and the germination of the seeds that they defecate. And so there are some very interesting things because you can see that they prefer the cultivated fruit. So it's very good for them. They survive. But this may have a bad effect in the long term if they are functioning as spreaders of invasive species, that they are facilitating the invasion of other habitat patches with these species. And if these species change also, if they are preferred by other pollinators and dispersers that stop eating the native species and prefer the cultivated ones. So you may have a change in community structure that will disrupt all the system in favor of the cultivated ones that are sweetest, are juiciest. And so we can try to make some of these things. The advantage of the cultivated ones is that they normally grow much faster. We have technology, but we need to be aware and careful about these consequences. And we can also try to promote stepping stones between isolated habitat patches so that individuals can move without being threatened by roads, by domestic dogs and other threats that they face, and normally that they die and do not make their way to other forest fragments to breed there and so maintain gene flow. So it's like depending on the degree of habitat fragmentation, we should only not mess with the monkeys, but depending if it's a very fragmented and if isolated habitat patches, we should promote this reconnection and increase in size and so on.
Okay, thank you for this answer. Actually, about consequence, I have myself a question, and especially regarding the publication of Janayana about supplemented howler monkey. So, like, of course, due to the urbanization, we can think that they have more access to, and especially in garbage, for instance, on very industrial and transformed food. And the fact is that we know that for human, the apparition of this kind of food also is correlated with the apparition of a lot of disease and stuff like this. Do we already observe, and are there already some studies showing the same kind of correlation between the consumption of this kind of food and the apparition of new disease in these monkeys?
I don't remember to have read anything about that. We are now knowing some differences in coloration, some of the problems that may be related to pesticides. But actually, howler monkeys do not go to the garbage. If they were capuchin monkeys, this would be a very likely behavior. But the howler monkeys, no. But one of these groups that Janaína studied, and she will study again, she's beginning to study again in her PhD, they do eat industrialized food. And this is one of our interests, to see if it changes their microbiota, and because they are eating very different food. And we are going to try to collect urine to see other markers of health. They are apparently fine. They do not show any external or behavioral sign of disease, and so they seem to cope very well. But it's also related with time. We don't know if the time frame that we had available for these consequences to happen is already sufficient to show a bad consequence. So as far as we know, they do very well, even when we thought that they wouldn't.
Okay, interesting. And I have a last question that will be more regarding all the cognitive part at the beginning. Like working with cognition with bees and fish, I often face the issue of motivation with food during the experiment. And I was wondering, how do you design your study just to be sure that you keep the motivation of the monkey during the full time? Because, like, I guess they are not eating as much as other kind of animals.
Well, this is also a very good question. First of all, we normally run these studies during the dry season, where the food availability is lower. But this is something that does depend on the monkeys. And this is a challenge of this kind of studies. Because sometimes, depending on what is available of wild foods, they will prefer the wild foods. Well, bananas for marmosets and tamarins, they normally don't know the bananas, but once they learn to eat, it's a lot of food together. So it's an easy food to get, and they really like, and even when you trap them, it's it's a stressful situation. You trap them, you take your hand, you anesthetize, you mark, and then you keep them some hours until they recover, and then you release. So you could think that they won't go back to the trapping station so soon. They are there next day. They say, no, I don't want to trap you. I want to trap the other species. Go away now. They are there eating again. And so it all depends on the reward. And they say, no, that's fine. Maybe they like the drug. and this is really dependent on what is available around, because we made actually 2 studies with capuchin monkeys here in the city, and we studied them because the person used to feed them, to supplement them with food. And then we did a study that worked quite well. And then the same student went to the PhD, and her project was to see the behavior of the monkeys with some manipulation of food amount and other. We increased the number of challenges to study social foraging. What happened at the time, everything was going kind of well, until somebody within the home range of the monkeys, that is, capuchin monkeys use much larger home ranges, started feeding them more. We didn't know the people. And so they stopped going to our feeding station. They, instead of going every day and rarely missing a day without visiting the station, they started going one day and missing 5. And it couldn't work for our experimental design, because we needed them to remember and to go back. And so 5 days is maybe too long. And then she needed to change her thesis project, like almost after 1 year of field effort. So it's, and we normally do in a time when they do not have that much food available.
Okay, very interesting. Thank you, Julio and Karin, for this really amazing talk. Like, I think we're done for today. Like I said, you can send us the other video like this. We will publish it, and I'm sure that people will ask questions also later. That's the point with all these conferences, that because we are trying to make it accessible for every time zone, like some question will come later because people will watch in replay. So yeah, thank you. Thank you very much. So I think the live is done for today. I will just remember for the audience that we will have another seminar coming the 27th of August. And also as a big reminder, like the 18th and 19th of November, we have the ABL Annual Online Congress that is completely free of access. So like, I invite really everyone to just communicate about it. It will be a 2 days conference with 4 plenary speakers and 12 short talks. So yeah, thank you everyone, and thank you, Julio and Karin.
Thank you everyone. Thanks again for the invitation, and good night, good afternoon, good day, good morning for everyone.
Yeah,
bye-bye.
really thank you too for being there.
Yeah, bye-bye.
Bye.
Bye.